Alright guys, so I put together a video here to cover the first part of this PowerPoint. Again, I'm sorry to juniors. I know it's not your fault that you weren't in class Monday, but to try to keep us kind of on pace, I thought this would be the best route to go. This is really a lot of review stuff. It's stuff you've heard before. If you take a look at the chapters and you take a look at this little video and the slides, I think you're going to be set. And then the latter part of this, we'll get into some more things that are kind of newer. You know, chapters 11 and 12 is all about cellular genetics, which another way to put that is patterns of inheritance. The way that we inherit our various characteristics. So we want to look at how those characteristics come from your parents, how we apply probability to that, which is what Gregor Mendel did, and then later on we'll get into more about how genes interact and lead you to the traits you have. Most of us kind of resemble our parents, but we don't look exactly like our parents, and that's because of the genetic variation we talked about last class, combined with the fact that you are still getting their genes in some combination. So, for instance, here's my wife and I and my brother, who's not really that short. He's just leaning down because it was funny. And these are my wife's parents. And so, particularly her father, she bears some resemblance, but she doesn't look just like them. And then here are my parents. And I think I resemble my parents a lot more than my brother does, but my brother and I look very different from each other. Then you look at Charlotte here, the cutest ladybug ever, and this is her little buddy Marshall, who's a friend of ours. And Charlotte, at this point, probably looks the most like her mom, maybe. But she's really got a combination of everybody's genes that are on this slide. So it'll be interesting to see when she gets a little bit older who she kind of looks like and if we can see characteristics of all of us in her. Okay, so I think this is the most awesome picture ever. Here's Gregor Mendel with his pea plant. And here's all Gregor Mendel's monk buddies. And what you have to think about is putting yourself in this picture, right? You know these guys, like you guys now, right? Here's the pose that our high school girls currently throw, right? Hand on the hip. And, these guys are sitting around like, how can I look cool? Like, look at this guy. I mean, this guy thinks he's awesome. And this guy's real contemplative, right? He's looking off in the distance. This guy's trying to look tough. These guys trying to look like they don't agree with Gregor Mendel. I just think this is an awesome picture. Look at this guy thinking with the finger on the chin. There's just so many awesome things about this picture. I just don't even know what to say. I felt bad because I marked in front of that dude's face. Now, my expectations on this stuff is that you remember Mendel, so you know that stuff. You can look at the book and fill yourself in if you need to. And you know those basics. He used pea plants. He did this. He did that. So that way we can use our class time to hit some of those basics that are most important, but then also look at some of those more detailed things. Okay, so here's some vocab, super fast, right? Remember, allele, different types of genes. Locus is your location of a gene on the chromosome. So here they're showing you we got two different alleles, purple and white. The locus is right here. Right? This is location on the chromosome. Gametes are those sex cells. And then trait refers to those characteristics that we see in you. So for this, you've got a flower-colored gene. So the trait we're talking about is flower color. You got two different alleles for it. Also remember these major terms. Phenotype. Remember, phenotype excuse me, is the physical expression of your gene. So that's like green or white or tall, while genotype is the actual genes. So you write that with letter designations. Right? Big G, little g gives you a green phenotype. Then remember you got dominant versus recessive. We'll talk about that more here in a minute. Dominant traits show up and kind of overshadow recessive traits. You use capital letters for dominant and you use lowercase letters for recessive. And then you had these terms, right? Homozygous and heterozygous. Homo meaning same. So homozygous dominant would be you have two dominant alleles. Homozygous recessive would be two recessive alleles. And then heterozygous, hetero meaning different, means you have two different alleles, one dominant, one recessive. A couple more terms. True breeding means that over several generations, an organism will only be giving rise to the same trait in its offspring. So if we see a true breeding plant or a true breeding organism, that is a pretty good indication that it's homozygous because we keep getting the same thing over and over again. When Mendel started with his parental or parent P generation, he used true breeding plants, ones that had been purple forever and white forever, or at least for many generations. Then he did what's called hybridization. He took two plants thought to be true breeding and he crossed them with each other to form a hybrid. Now, today we have the term hybrid, and we also know that that probably means heterozygous, right, if it's a hybrid. Then if you cross two hybrids, you get a different outcome, and so we'll get to that more later too. Okay, a little more vocab still. When we do a test cross, this is where we cross a recessive organism, little g, with a mystery dominant organism, meaning that it could be big G, or could be big G, little g. By doing this, we can determine whether that mystery organism is homozygous dominant or heterozygous, because we'll get a different outcome in the offspring when we cross homozygous recessive with each of these guys. Last couple things, remember a monohybrid cross is where you're looking at one trait, so it's a simple 2 by 2 Punnett square. A dihybrid cross is where we're studying two traits at the same time, so that's where you had to do a 4 by 4 Punnett square. Okay, two laws that came from Mendel's early work. The first one is the law of segregation, and this law states that every individual has two factors, which we now call alleles, for each character, which we now call traits. And what Mendel said is that these two alleles are going to segregate or separate when we form our sex cells, we form gametes. So in other words, in meiosis, you're going to separate the two alleles for a trait, and that's because you've got one allele on one chromosome and the other version of that gene on your other homologous chromosome. Now, because these two things will separate, each gamete will carry only one of those alleles. So fertilization will give one of two different alleles from each parent when you form your new offspring, giving that offspring two alleles total, but one that came from each parent. And those two alleles and the parents to start with will separate from one another. Now, he also had a law of independent assortment, and this is different because in the first one, law of segregation, he's looking at one gene, right, with two different alleles. Here, he's talking about different genes, and he says that different genes will also separate independently from other genes, meaning that in your kids, you can get any possible combination. Sort of the classic go-to way to put this is that just because you have blonde hair does not mean you have to have blue eyes. All right, now that's a grossly oversimplified way to say that, but that's what we're going at here is that you don't necessarily keep two traits together all the time. Those two things can separate. So in the law of segregation, one gene, but alleles separate. Right? In the law of independent assortment, two genes, and they also are inherited separately. Now, the way you would figure this out is with dihybrid crosses. And you also want to be careful here because we do now have two types of independent assortment. We have Mendel's independent assortment of genes, and then we had last class the independent assortment of chromosomes that we see bringing us variety when we do meiosis. Okay, so to determine your gametes, what you do is you set up a Punnett square, right? So when you set up the two sides of your Punnett square, what you're really doing is you're considering what possible gametes could this organism form. So what we look at is each gamete will carry one allele, so that gamete will never have more than one of the same letter. So if a plant has the genotype GG, and we stick with just this one single gene we're looking at, then it could form gametes with a big G, and it could form gametes with a little g. And if we want to be more specific to meiosis, right, we're going to form 4 cells in the end. So we have 2 chances to form big G and 2 chances to form little g. Now, in a dihybrid situation, it gets a little stickier, right? Because now we have to do a little extra work. Still, each possible gamete will only get one of each letter. So if you have this big G, big R, organism for green and round, you can get 4 possible gametes. You can get the big G with the big R. You can get the big G with the little r. You can get the little g with the big R. And you can get the little g, these are getting ugly now, with the little r. And again, this is what you'd put across the top of your Punnett square if you're making a Punnett square, or if you were doing this one, you'd just put that right across the top of your Punnett square. So you're always really just looking at what kind of gametes am I making. Punnett squares are awesome, right? They're a helpful way to diagram the outcome of a cross and to figure out the probability really without looking at a whole lot of math, right? Please always feel free to draw Punnett squares. If you think it'll help you on a question, then totally draw a Punnett square in the margin. And we'll just assume at this point that you do know how to cross and drop and do a Punnett square like you did all through freshman year and probably before that too. The only way to really get comfortable with Mendelian genetics is to work on practicing Punnett squares, and you did that a lot in previous classes. This is the main reason why I gave you that practice packet, so that you have plenty of problems to take a look at and try your Punnett square skills. Something interesting to notice is that the AP exam is probably not going to ask you to draw a Punnett square. What they more expect is that you realize when you need a Punnett square to answer a certain problem, like a word problem or a free response question. So what this means is they're not looking to grade your Punnett squares necessarily. They're just looking to see, did you get the right answer to this bigger question? That means you don't have to always make a Punnett square. So as we'll talk about later and you'll see on that practice sheet, there's sort of shortcuts you can take sometimes, so you don't have to sit there and actually fill out a Punnett square, especially if it's one of these big 4x4 ones. But again, please do make Punnett squares whenever you need to. So if you need to make them in the margin on the multiple choice part, do it. If you need to make them for a free response question, do it. Just realize it might not get you points, but it will probably help you get the right answer. Okay, so the last thing we want to talk about in this PowerPoint, or this little segment of it, is non-Mendelian inheritance. And these are the same things you learned in your freshman year. There are 3 major things that we see that do not follow Mendel's laws of segregation and independent assortment, and therefore were not really well reported or explained by Mendel. This is where alleles are not completely dominant or recessive, a gene has more than 2 alleles, or a gene produces multiple different phenotypes. In all of these cases, these are ideas that would have supported Mendel's competitors. People who disagreed with Mendel would have cited these as cases that Mendel was wrong. As it turns out, it wasn't that Mendel was wrong, it's just that these are a little bit more complicated genetic cases than the pea plants he was looking at. Okay, so the first thing is this idea of incomplete dominance, right? The normal condition is complete dominance, right? You got a dominant, you got a recessive, and the dominant overshadows the recessive, that sort of idea. Incomplete dominance is the idea that neither allele is totally dominant over the other, which gives you sort of an intermediate phenotype when you have a heterozygous organism. That classic example that you've learned a couple different times now is red, white, and pink flowers, particularly snapdragons, that we see in this picture right here. So red and white are both, the color here is an incompletely dominant trait. So if you cross a red flower with a white flower, you get a pink flower, the heterozygote that has both this R allele and this W allele. Notice that they don't write it like this, right? They don't just put the R's and the W's. That's because this is the same gene. Alright, so since it's the same gene, we have to use a consistent letter, and that's, I don't know why they chose C here, but that's why there is a C with a superscript R, because C with a superscript R is the same gene as C with a superscript W, it's just the different alleles. Now, a lot of the people who were against Mendel said that, hey, this is an example of blending inheritance, which is what they thought happened. But what we can now show is it's not blending inheritance, because if you then cross the pink flowers with each other, you don't get all pink or other shades of pink. You can go back to having red, you can go back to having white, or you can get pink again. Now, another idea with this dominance stuff is the idea of codominance. Codominance means that both alleles affect the phenotype in separate, distinguishable ways. So it's not that you get an intermediate if you're talking about codominance. Intermediates show up with incomplete dominance, so red, white, pink. With codominance, it's kind of like you got red, you got white, and the offspring would be both red and white, maybe in different areas. A good example of this is a way that we can do blood type other than the really common way we do blood type that you guys are probably familiar with, and that's the MN blood grouping system. Now, all the blood typing things look at proteins, antigens, on the surface of your red blood cells. There's a group of these which we categorize as either M, as in monkey, antigens, or N, as in, Neverland, antigens. If a person has the genotype MM, they have all M antigens. If the person has the genotype NN, they have all N antigens. But this is a codominant trait. So if someone has genotype MN, one of each, then that red blood cell is going to have both M antigens and N antigens on its surface. So that's what codominance means. Okay, so this slide brings up something that's definitely on the AP level, which is what the concept of dominance actually means, or where it comes from. You see, when something's dominant, it doesn't mean that allele suppresses another allele or overtakes it or anything. When something's dominant versus recessive, what that really just means is there's a molecular difference in that cell, and therefore in that organism, that comes from gene expression. So what do I mean? Let's look at peas. Okay, peas can come in round or wrinkled, as Mendel studied them. Round is the dominant condition, wrinkled is the recessive condition. Now again, this doesn't mean that round alleles knock out the recessive ones or anything like that. Here's what this means. Round is dominant, and in those cells, a dominant gene codes for an enzyme. How about that? It's all about enzymes, right? That forms a particular type of starch. A wrinkled pea is what we call recessive. In a wrinkled pea, it lacks that enzyme. In other words, the other allele, the recessive allele, doesn't code for that enzyme, which means you don't make as much of that starch, which means you don't have an influx of water by osmosis, and the seed isn't filled up. All right, now the first thing is, I have a typo in your set of the notes. It says leading to influx. It's really less influx, right? Less water flowing in. But now we're just talking about cells, right? If you have a cell and it has a whole bunch of stuff inside, the water rushes in and the cell becomes turgid in the case of a plant, or the cell swells. So if you have the dominant allele, you make that sugar that's in there, water rushes in, and you end up being round. The seed is taut, right? If you have the other allele, you don't make the sugar. The sugar's not in the cell at high concentration. Water does not rush into the cells, and you're wrinkled and shrivelly. Right? Now, only one round allele is enough to make some sugar and keep the seed, swollen. So that's why the round allele is dominant. You only need one of those alleles to make enough enzyme to make this happen. But if you don't have that allele at all, if you have two of the recessive allele, you don't make the enzyme, you don't get the sugar, you don't get a swollen seed. So that's what it means. It's not that dominant is better than the other thing, it's just a difference biochemically. Now also notice that dominant does not mean a more common trait. We'll get into a couple examples of this later, but in humans, having 5 fingers is recessive, and yet we almost all do. You also might remember from freshman year, being a dwarf, really short, is dominant, but most of us aren't, right? Another instance where we don't follow a Mendelian pattern is that some genes will actually have more than two alleles. So instead of just having like a big T or a little t, you've got more options to choose from. Now, each organism is still just going to have two alleles, one they got from their mother and one they got from their father, but they have more alleles to choose from. And classic example of this in humans is the blood typing system we're all more familiar with, which is the ABO blood typing system. In this system, you can have an A allele, you can have a B allele, or you can have an O allele. Now, this means this is an example of multiple alleles, so you have three to choose from. It's also an example of codominance, because if you notice here, A and B are both dominant to the O allele, but A and B are actually codominant to each other. So if you have both, you get both. Last thing for this little video is another case of non-Mendelian inheritance, which is that a lot of times a gene will have multiple phenotypic effects. So what we mean is one gene might actually influence several different characteristics that you have, and we call this pleiotropy. So if a gene is pleiotropic, it means that it has an effect on more than one of your phenotypes. So for instance, since the time of Mendel, we've actually discovered that the gene for flower color in peas probably also contributes to the color of the seed coat. So not only will that gene help control what color the flowers are, but it'll also help control whether the seed coat is green or yellow or something of that kind. Now in humans, there's not a lot of examples that are well understood because, as we'll see next class, there's a lot of complicated stuff that we have to consider when we look at human genetics. But a lot of human genetic disorders will have several symptoms in different parts of your body. And these won't all come or stem from the same change in your phenotypes. It's actually that there's multiple phenotypes that are getting altered. An example of this is not a real good example, is that some diseases might give you a certain physical look, but they also might cause heart defects on the inside. Now those are because of two different kind of functional changes that are going on, but it all might come back to that same exact gene. Okay, so this is enough for now. We'll pick up here next class and move forward and hit the rest of this stuff about cellular genetics.